Hi, I'm Dr. Aisy Anaqi and I'm majoring in biotechnology. So today I'm going to be discussing about recombinant technology, DNA technology with my friends over here.
Hi, I'm Professor Dayana and I'm majoring in genetics.
Hi, and I'm Nornabilah. I'm majoring in genetic engineering.
Hi, my name is Professor Adriana Karmila and I'm majoring in microbiology.
This topic, let me tell you what recombinant DNA technology is. So basically, recombinant DNA technology means a method in which DNA molecules made in vitro with segments from different sources or by binding gene from different organisms. This recombinant DNA technology has been happening in our daily lives since we were little babies. One of the examples of recombinant DNA technology is vaccination, which DNA technology has been put in our body, and then heart attack treatment, cancer treatment, and many more.
In recombinant DNA technology, there is a few tools that we use. First of all is target DNA. Target DNA is a sequence of DNA containing gene of interest. The example of target DNA is gene encode for protein, which can dissolve blood clots, which is used in heart attack therapy.
So, for the second tool is a restriction enzyme. Enzyme which cut DNA molecules at the restriction site and produces short restriction fragments. Restriction site is the short base pair sequence of which is of 4 to 8 base pair recognized by a particular restriction enzyme. For restriction site is contain palindromic, where same sequence of bases is found on both DNA strands but in opposite direction. The example for restriction enzyme, which is first EcoR1 and the second is SMART1. Now I'm going to show the cutting process for target DNA. Right now, the first example for restriction enzyme is the E. coli. For E. coli, is produce the E. coli. This is for Caesar. We can assume as a E. coli, which is a EcoR1, something like that. And then for this is for target DNA. Right now, I was highlighted, which is this line, which we cut for this, for the staggered. Yes. Okay, right now I will show how to cut it. Just cut like this. It's very simple that you guys need to know how the process will be. You just follow this line and then yes, just like this, and then we spray it. Yes, that's why this target gene was made a staggered cut. Yes. For this cutting, it will produce a sticky ends, which is a single strand ends. For the second cutting process, which is example is a small one. This is here, small one, and this is a target gene. You guys can assume this is Caesar as a small one, and then this is target gene. For small one, is produced as Serratia mersensen and recognized by the basis sequence of, we could see that C, 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 and G, G, G. So the cut made between the adjacent, which is the C and G. Right now, this line I will cutting here, cutting like this, and then it will spray it, and then it will produce blunt ends.
Next, our third tool for our DNA recombinant technology is the DNA cloning vector. DNA cloning vector is a DNA molecule which can carry foreign DNA fragment into a cell and replicate it in that cell. DNA cloning vector should able to accept foreign DNA in multiple cloning sites, and also DNA cloning vector should able to replicate freely in the host cell.
The next tool is host cell. Host cell is a cell which receives recombinant DNA and allows the replication of recombinant DNA. Some of the characteristics are they are able to receive recombinant DNA through a transformation process. And the second one is to maintain the structure of recombinant DNA from one generation to another. And the third one is to amplify the gene product from the recombinant DNA. One of the example of hostile is bacteria. This is because it is common use and can be easily isolated from or introduced into a bacterium. It grows quickly, rapidly replicating any foreign genes they carry. Example for bacteria is E. coli.
The last tool is modifying enzyme. There are two types of enzyme. The first one is DNA ligase. The second type polymerase. DNA ligase join the foreign DNA fragments containing gene of interest in bacterial plasmid by utilizing the formation of phosphodiester bond to form recombinant DNA. The second is tag polymerase. Tag polymerase used in polymerase chain reaction (PCR) and replacing the DNA polymerase because of the temperature conditions of PCR. And it also add nucleotides to the 3' end of primers in order to produce new DNA sequence.
Today, my friends and I are going to be experimenting on Hazim's body by applying the recombinant DNA technology to help him with his problem due to his low metabolism rate, immune system, his body size, and many more. So we start off by doing process 1.
So this is first process. First process is isolation of gene. Isolate DNA from foreign cells containing gene of interest and plasmid from pentacells.
So for the second process is cut process. Right now in my hand is a cloning vector, which is I need to cut with this EcoR1, which is from a restriction enzyme. I cut this and it will produce sticky ends because it become like this. And then for the next cutting is a small one, which is I cut like this and become like this. So we call it as blunt ends, which is the produce blunt ends.
Next is the process of insertion. The insertion process requires that a gene of interest, 2 types, and our cloning vector. SMA1 and EcoR1, which we use DNA ligase as glue to our DNA and into our cloning vector. Okay, so this is the vector that have been recombined with our DNA gene interest using DNA ligase for staggered cut, and this is our DNA recombinant that is in DNA ligase also as glue to glue our DNA gene of interest into the vector.
So this is a vector contained with DNA recombinant. We have 5 possibility. One bacteria containing DNA recombinant with staggered genes. One bacteria containing DNA recombinant with blunt end. And next, there is a possibility to have one bacteria that contain no DNA recombinant but have antibiotic, and no DNA recombinant with no antibiotic, and last one is no vector.
The last process is screening process. Screening process is to identify bacteria that carries the recombinant DNA from all the 5 possibilities that we get from the amplification process. So these are the probability of the bacteria that we have no idea if it has recombinant DNA. But if we want to know how to find the recombinant DNA, we must use the blue-white screening technique. We place this petri dish with agar that contain antibiotic, amplicin, and X gel. And then after that, we're gonna put this into an incubator and leave it overnight, and let it grow for a while. And after few hours, we're gonna get a result like this. Okay, the bacteria that does not grow is the bacteria which has no vector. These blue colonies are the plasmids that has no recombinant, and the white ones are the ones that has recombinant plasmid. Recombinant plasmids of this blue contain intact like Z, which can produce B-galactosidase and can hydrolyze X gel, while the white has recombinant plasmids that do not contain intact like Z, but it cannot produce B-galactosidase and cannot hydrolyze X gel.